We've been talking about reproductive isolation and speciation in plants. And in the first half of this lecture, we talked about why are angiosperms more diverse than other plant groups. And we've been talking about the roles of reproductive isolation and speciation with regard to pre-zygotic and post-zygotic isolating mechanisms. We're still talking about pre-zygotic isolating mechanisms. We just talked about ecogeographic barriers, and we talked about differences in timing of pollination as a pre-zygotic barrier. We're now going to talk a little bit about pollination syndromes and attracting different pollinators as a pre-zygotic barrier. After that, we'll talk about post-zygotic isolation and adaptive radiation. So here again is that list of pre-mating barriers. We've talked about geographic, ecogeographic, and temporal isolation. So we're on to floral isolation. Floral isolation can occur if different incipient species, so populations that are undergoing speciation, if they attract different pollinators, or possibly if they attract the same pollinators, but they place their pollen on different parts of the pollinator's body. So how does floral isolation arise? Typically, this is going to start with some other barrier to hybridization, for example, a geographic barrier. So let's imagine that we have a population over in this area, and it has flowers that are adapted to, let's say, bees. And then some seeds manage through some chance event to disperse across a mountain range. And now we have those same flowers on the other side. Once we have that geographic separation, the next step is for there to be either evolution or coevolution between the plants and pollinators in different areas. So coevolution specifically means that traits of both the plants and pollinators are undergoing selection. Alternatively, we could simply have evolution without the co, which means that the plants are responding to the pollinators in the two areas without the pollinators themselves changing. Either way, in our example, let's imagine we've got bees on this side. So this is a bee. And over here, we have hummingbirds. Imagine that's a hummingbird. Here's its eye. And so the traits that are conducive to get hummingbird visits and for them to be successful pollinators are different than those for bees. We'll talk more about that soon. So once these flowers are now in an area with hummingbirds, there will be selection for changes in floral traits. Maybe the change in flower color to red and maybe a more tubular floral shape. Maybe bilateral symmetry would be common traits for a hummingbird flower. Something like this. Now, let's imagine we have now some divergence. The next step to make sure that this actually leads to complete speciation is remixing. So now let's imagine that some seeds from the right-hand side, where we have this new hummingbird flower, get dispersed back across our mountain to the initial population where we had these more actinomorphic flowers without the tubes. We now, to complete speciation, would need a process of reinforcement. What this would mean is that where the two species now coexist, there's even stronger selection for them to develop traits that minimize the sharing of pollen between the two incipient species. So during this time when they were separate, maybe we got the accumulation of selection for different traits, floral traits as well as other traits. Maybe we also had, through genetic drift, accumulation of changes in the two species. What that means is that when they get back together, if they ever do form hybrids, so I'll just draw a hybrid forming between them, then either because of the floral traits or because of those other traits that they've accumulated, the hybrid now has low fitness. Draw a frown face. That means each time they use their pollen or use their ovules to make a hybrid, they're kind of wasting it because they're making a plant that's not going to have high fitness and not going to leave behind many offspring. As a result, in this area where both species now occur, we get selection for even more dramatic differences. So this tubular flower becomes really tubular, and maybe it becomes really zygomorphic instead of just a little bit like it was before. And this round flower now maybe becomes even rounder and with even less of a floral tube, just to make sure that hummingbirds never visit this flower and that only hummingbirds visit this flower. So the differences that initially occurred in allopatry or in separation have now been reinforced and are exaggerated. This means that gene flow is even further reduced compared to what it was when they initially came back together. And in the example we've been talking about, the plants have evolved different pollination syndromes, different suites of traits that attract different sets of pollinators. So let's look at an example of this. So we are going to look at an example of reinforcement for character displacement in orchids. In other words, we are going to have this process where selection has led to exaggerated differences between two species to minimize pollen exchange. And this has led to a character or a trait becoming even more different. That trait then has been displaced. The example is in orchids. Orchids are the second most diverse plant family after Asteraceae or Compositae. And in this example, this is not a reinforcement for different pollinators. Rather, it's reinforcement for where pollinators put pollen, or rather where flowers put pollen on the pollinator's body. We have two species. We've got Planthera chlorantha, which is pictured at the top, and we have Planthera bifolia, which is pictured at the bottom. And in this example, there's a difference in the width of the sticky parts on the pollen. So the floral structure is not identical to Asclepiadaceae, but it's similar in that there are big sacs of sticky pollen that adhere to pollinators. And then the sticky part, the viscodium, is in this case labeled here and here. So you can see that the sticky parts are more narrowly spaced in bifolia and more widely spaced in chlorantha. What this means is that when a pollinator puts its proboscis into the flower, then in bifolia, the pollen is going to stick near the base of the proboscis. Here's the pollinator's eyes up here. In viscodium, with a wider mouth and wider position of the viscodium, when the pollinator puts its mouth in, then it's going to get its proboscis further in before the viscodium contact the pollinator. And so they're going to stick up closer to the eyes. As a result, the pollen's on different parts of the pollinator. That means that even if a pollinator is visiting both of these species and not differentiating between them, the pollen that's picked up from bifolia flowers is also more likely to be dropped off on other bifolia flowers. Similarly, pollen that's picked up by chlorantha, where the pollinator puts its head further into the flower, is more likely to be deposited on other chlorantha flowers. If a pollinator's visited, let's say, chlorantha, and it then goes to visit a bifolia flower, it's going to put its proboscis into the flower, but only up to about here. that means this pollen near its eyes is not going to get transferred. But when it goes back to a chlorantha flower, it's going to put its head further into the flower, let's say up to here, and there will be the chance for this pollen near its eyes to be transferred. So we've had character displacement in pollinator placement between these two species, allowing them to occur in similar areas while minimizing exchange of genes. I'm going to skip quickly through a second example of pollen placement from 4 different species on hummingbirds. The general idea is the same, which is that if different species have different shaped flowers, then they will put pollen on different parts of the hummingbird. And so we've got 4 different species shown here. And what we can see is that depending on the different plant species, pollen is distributed on different parts of the pollinator, either on the bottom of its beak or the top, and either near the tip of the beak, as in this example, or further near the base of the beak, as in this flower, and either on the top of the beak, as in this flower, or on the bottom of the beak, as in this flower. So different species then can use the same pollinators and still minimize exchange of pollen. This example is not closely related species as it was in our orchid example, but the general idea of partitioning a pollinator so that different species can use it while minimizing exchange of pollen is the same. So let's move on to an example where we will look at pollinator shifts. So this then is an example not of using the same pollinator and putting pollen on different parts, but rather switching pollinators completely. We've looked at portions of this example before. This is pollinator shifts in columbine. And remember, I first showed you this figure when we were simply talking about ancestral character state reconstruction. And we said that we could use the principles of parsimony to infer what were the likely ancestral character states. And we said that flowers can have different colors, purple, red, or white. Let's now add to that red flowers tend to be pollinated by hummingbirds. So this got covered up, but that's red. The purple flowers tend to be visited by bees, and the yellow flowers tend to be visited by hawk moths. So now if we look back at our ancestral character state reconstructions, we're not just looking at flower color, but we're also looking at what the putative or the likely pollinators are. And what we can see here is evidence for several occasions where there has been a switch in the kind of pollinator. And we can see that one frequent shift is from bee pollination to hummingbird. So here's one example right here. Here's another example from bee to hummingbird right here. We never get shifts back. Once they go to hummingbird, they never go back to bee. We also see even more examples of shifts from hummingbird to hawk moth. So that's going from an ancestral character state of red to a character state of yellow. And here's one example down at the bottom. Here's a second example of that shift. That's 2. Here's a third example where the ancestral flower had hummingbird pollinated traits and it shifted to hawk moth. And another example, and you can find some more on here too. So a lesson here is that we do sometimes get shifts coinciding with speciation between different kinds of pollinators. We also see that there are some trends in the direction. For example, we get shifts from bees to hummingbirds, but not back. We also get shifts from hummingbird to hawk moth, but not back. And we don't have any evidence here of a direct shift from bee to hawk moth. Obviously, if plants are using different pollinators, that will minimize exchange of pollen and hence exchange of genes between those two different incipient species. Let's talk more generally about pollination syndromes. Pollination syndromes are suites of traits, so combinations that allow the plant to both attract and benefit from whatever their most important pollinator is. Traits tend to work together. There are some traits that are very good for some pollinator types and other traits for others. And so we see repeated evolution of similar combinations of traits. And we won't go through them all, but I'll give you some examples. One classic combination of traits is good for moth pollination. Flowers tend to be large and white. We can think about why large and white. Obviously, it's dark out, so it's hard for pollinators to see flowers because moths tend to visit at night. So by having large flowers, they're easier to see at night, and white is the most distinctive color at night. And so again, this is making it easier for moths to see. Now, moths still aren't going to be great at seeing in the dark, and so these flowers are also highly fragrant. It gives the moths a different sense with which to locate flowers. Moth-pollinated flowers have long nectar-filled tubes. The reason for this is that moths have long tongues. The tongues roll up. And so it's a way of keeping all of that nectar isolated so other pollinators with short tongues will not be able to access it. And they need a lot of nectar because moths spend a lot of energy pollinating. They pollinate while they're flying. They do not land on flowers, so that takes extra energy. They're pollinating when it's cool outside at night, and so they need to spend extra energy to be able to maintain their flight in the cold conditions. Moths don't want to land on flowers. This is partly because at night they can't see if there's a spider or other predator hanging out on the flower. And so instead of landing, moth flowers hang downwards, like so, and the moth hovers below. It doesn't get too close. It extends its proboscis, and I should have drawn this as a tubular flower, so I'll add a tube here. And it accesses that nectar that's in the base of the tube, staying far enough away that if there was a spider sitting on this flower, try to draw 8 legs there, then the moth would be far enough back that the spider would be unable to catch it. So large white nectar-filled flowers with fragrance and a pendant or downward orientation is typical of a moth-pollinated flower. Let's contrast that with a bird-pollinated flower. Birds are especially good at seeing red. That means red flowers are often associated with bird pollination. There's an example here of a yellow flower, so it's not always red, but it's the most common. And we saw that back here with the columbine example. These flowers were red and secondarily yellow for hummingbird pollination. Again, we're seeing that red and yellow in these examples. Like moths, hummingbirds are not going to land when they feed, so they are fluttering. And also because hummingbirds are, what's the right term? Endotherms, I guess. They are consuming a lot of energy. So they need to have a lot of nectar in order for visiting flowers to be worthwhile. So hummingbird flowers have large quantities of nectar. They have red or secondarily yellow flowers. They are narrow, and the reason that they're narrow, if we look at the face of a hummingbird, it wants to be able to watch for predators while it's feeding. And its eyes are, maybe moderately widely spaced, but not extremely widely spaced. So the best way for it to be able to watch its environment while it's feeding is if the flower it's visiting has a very narrow corolla. I'll draw it like that. So this is the flower I'm drawing, and we're looking at the face of the pollinator behind it. You can see if it's a narrow corolla, then the pollinator is able to see what's behind that flower and watch its environment even while it's feeding. hummingbirds and other pollinators have very long, narrow beaks, and so that explains the narrow tubular flowers. Again, this is a way of keeping other kinds of pollinators from accessing that nectar and only allowing those that either have a long proboscis or a long beak to be able to reach inside and get the nectar that's at the very base of these tubes. So we've talked about moth and bird pollination. Let's talk about fly pollination. Flies tend to eat carrion or dead decaying things, and so a flower that's fly pollinated will tend to be maroon or brown in color, kind of like a decaying animal, and they will tend to smell badly, again, like a decaying animal. So think about skunk cabbage and the stink that comes with it. Or this is a species of Trillium, and I believe that this goes by the common name, among others, Stinking Benjamin. And I'd appreciate if that does not become my nickname. But it's so called because it has a smell, again, of carrion or dead animal. I think I misspelled fowl here. I think fowl in this sense should have a U. Finally, we've seen some examples of wind pollination. If a plant is wind pollinated, it's not trying to attract pollinators, but it is trying to get its anthers and its styles out into the wind. So in this case, petals and sepals are going to get in the way, and they tend to be absent or highly reduced. There's no reason to make scent or nectar if the plant is not trying to attract pollinators, and so those traits tend to go away. But what's important for a wind-pollinated plant is to make a lot of pollen, because it's a very inefficient mechanism. Most of the pollen seems to end up on my car or on my deck or on my bed if I have the windows open. So not much of it is going to reach flowers of the same species. Plants compensate for this by simply making a lot more pollen. So we've seen examples of this, and the two that come most immediately to mind are catkins in Fagaceae and Betulaceae, and even those catkin-like female inflorescences in Betulaceae, which hang out into the wind. Remember, even the female flowers in Betulaceae were on catkin-like inflorescences. Draw some styles on here. And this hanging out in the wind is going to allow them to capture pollen that's blowing by. So now we've talked about 4 pre-mating barriers to isolation: geographic, ecogeographic, temporal, and floral. Let's move on and think a little bit about post-mating pre-zygotic barriers. This is a barrier that occurs after pollen lands on the stigma, but before the pollen tube has grown down and allowed its sperm to be released to fertilize the ovule. I will show you an example from my own research that I did as a postdoc with Linda Delph at Indiana University. And in this research, we looked at post-mating pre-zygotic isolation in Selene. Selene's in Caryophyllaceae, and you should be able to recognize these flowers as such, shown here. These two species, Selene dioica and latifolia, are interfertile. If you cross them in the laboratory or greenhouse, they will easily make large numbers of seeds, and those seeds are viable. In nature, where these two species have overlapping ranges, we find some hybrids, but not large numbers of hybrids. So what we're interested in is what is preventing these two species from hybridizing in nature. Now, there's some ecogeographic differences, and there's some phenological differences. So post-mating pre-zygotic isolation is not the only thing going on, but it does seem to be a contributing factor. And what we looked at specifically is, first, how well do pollen tubes grow in flowers of the own species compared to flowers of the other species. We tested this on both species. So we had dioica as the plant receiving pollen and latifolia as the plant receiving pollen. And we applied dioica pollen, which is the white bars, and we applied latifolia pollen, which is the dark bars. So if there is a post-mating pre-zygotic isolating barrier of different pollen tube growth, we would expect the own species pollen to always grow better. And that's what we see for dioica. You can see that on dioica flowers, dioica pollen tubes elongate longer than they do on latifolia flowers in the same length of time. I don't remember, but maybe after about 4 hours. And in latifolia, we see the same pattern. Latifolia pollen tubes grow faster and hence reach a longer length per unit time than do dioica pollen tubes on the same flowers. What this means is that if a pollinator arrives with a mix of both pollen types, the pollen from the own species is going to be able to grow pollen tubes faster and hence achieve pollination more quickly or fertilization more quickly than pollen from the other species. This means that the number of hybrids will be reduced compared to if they're pollen all elongated pollen tubes at similar rates. When we look at actual formation of hybrids, what we see is that on one species, if we put on a mix of pollen, and so this is the proportion of heterospecific pollen that we put on, in other words, the proportion of pollen from the other species, we see that if we put on a mix, then we get a lower proportion of hybrids forming than we would expect if the pollen did equally well. So these points are all below this 1-to-1 line, which would mean equal success for both pollen types. So this is an example then of a post-mating pre-zygotic isolating barrier. Foreign pollen does not sire as many seeds as pollen of the own species. For the other species, when we put on foreign pollen, there wasn't really a disadvantage. That foreign pollen did as well or even very slightly better than pollen of the own species. And so these results partly agree with what we saw for pollen tubes. They agree for one species, and for the other species, there wasn't a strong disadvantage. So we have evidence here for at least one of these two species that there's a post-mating pre-zygotic barrier in terms of being able for pollen to fertilize seeds. So now we need to move on and talk about post-zygotic barriers to hybridization. So one post-zygotic barrier, before we get to the slide, let's just talk a little bit. One post-zygotic barrier is that hybrids might simply be less fit. If we imagine plants growing on a mountainside and we have the upper population and the lower population, if the upper population has traits good for this high elevation and the lower one has traits good for this low elevation, and we get the formation of hybrids, if those hybrids don't do very well in this upper area, just draw a frown, and if hybrids don't do very well in the lower area either because they're not really well adapted to either area, then those hybrids will tend not to leave behind many offspring, and these two populations could maintain their separate identities and continue speciating. So that would be a post-zygotic barrier based on selection for different traits. It's post-zygotic because it has to do with what happens after fertilization. It's a barrier because hybrids are not doing well, and hence they are not being selected. They're not passing down many offspring, I should say. And that's preventing hybrids from persisting in the area. We're now going to shift gears to what's on the original slide and talk about post-zygotic isolation that can happen even through non-selected changes. So this would be processes like genetic drift. And And what we know is that genetic changes can accumulate over time, such that when the two populations that have these changes are reintroduced, if they do cross-fertilize, then the offspring may not be viable. They might have a genetic problem that prevents those seeds from reaching maturity or germinating, or the germinated plants from surviving. So there's a paradox here. Alleles that are going to eventually lead to incompatibility between two populations must have evolved within one or the other population. However, if those new alleles are incompatible with other genes that would not get passed down, then they couldn't have ever increased the fixation in the population. So how do we get evolution of a gene that's incompatible if both of these populations are starting with the same genotypes? So on initial sort of examination, this would lead to the conclusion that it should not be possible for populations to become fixed for alleles that are going to cause incompatibility between populations. Because that change must have happened somewhere, and that change must have caused incompatibility at the time. So this is a paradox. We know that it happens, but we also can explain based on these logical steps that it shouldn't happen. We do have genetic models that will help us resolve this paragraph. And Dobzhansky-Muller incompatibility is the solution that we think leads to the evolution of genetic changes that prevent hybridization when two populations come together. The general idea is that in each population, new alleles for different genes become fixed. And in each case, the alleles are compatible with the original genotype, but they are incompatible with one another. This is easier to see with the diagram. So we're going to think about two genes, and we'll just call them gene A and gene B. And we have some original population that has, let's say, the dominant trait or the dominant allele for both of these genes. Now we get separation. So some seeds are dispersed across a mountain range or across a large river or something like that. So the two populations are now separate. Initially, they both have the same original genotype because there hasn't been time for any changes. But over time, there's a mutation in this left-hand population, and a new allele, little a, arises. And let's just say by genetic drift, over enough time, it eventually reaches fixation in our, we'll call it population 1, the left side. In population 2, let me fix a mistake here. Let's make this big A. Okay. In population 2, initially the genotype is the same as the original population, but again, at some point we have a random mutation that creates allele little b, so recessive allele. And just by genetic drift, over time, allele B becomes fixed in population 2. Now let's imagine there's some event where seeds from population 2 get dispersed across the mountain and end up back in population 1. And as a result, we have hybrids form between these. And here's the genotype of our hybrid. It's going to get little a from pop 1, big A from pop 2, big B from pop 1, and little b from population 2. So it is now a double heterozygote. Now we have a situation where little a and little b are seeing each other for the first time. This is the first time they've been in a plant together. Now, allele little a does fine with allele big B, right? We have got plants here that have both, And we even have plants here that have both, where little a is being expressed because it's a double homozygote. So there's no incompatibility between little a and big B. Conversely, over here in population 2, we have the allele little b, the recessive one, being expressed. It does fine with allele big A. So no problems with this genetic combination. But now we have a new genetic combination. In this F1, there's still no problem because little a and little b, in my example, are both recessive alleles. So the phenotype here would be whatever phenotype's associated with big A and whatever phenotype is associated with big B. That's the same as our original population, and we know that big A and big B phenotypes are compatible with each other. But let's think about what happens when this F1 self-pollinates and creates an F2. So we can make that Punnett square. But basically, we're going to come out with a bunch of possibilities. We could end up with big A, big A, big B, big B. We could end up with big A, big B, little b. Well, of course that. And you can list these all. Who am I missing? Big, little, and... So in most of these cases, we are going to end up with combinations that are compatible. So we know big A and big B are compatible. They were in our original population. That's fine. Here, it's still expressing. Let's just make a list of the phenotypes. These are, of course, genotypes. So here, our phenotype is big A, big B. That's our original phenotype, so we know that's okay. Are they functional together? Yes. Here, our phenotype is still big A, big B, and that's true here. And so we know that all of these are going to be fine. Here we have a little a, big B. We know that combination works because that's the phenotype associated with population 1 right before they come together. So little a, big B is fine. Here we have phenotype little a, big B again. And it looks like I've left some off on the list. We must have some that are big A, little b. So we're going to have, for example, big A, little b. That would give us phenotype big A, little b. But that's the phenotype associated with population 2. So that's perfectly functional. So now let's focus on this phenotype. Little a, little b. If there's an incompatibility between the allele little a and the allele little b, now that's going to be expressed because we have phenotype little a, little b. And let's imagine that those two create inviable zygotes. This then is a non-functional allele. What this means is that, especially in the F2 generation, we are going to have some proportion of individuals, some proportion of seeds that become inviable. As a result, the F2 are going to have lower fitness on average than pure members of either population 1 or population 2. So when these come back together, this is going to mean that there is going to be strong selection to avoid intermating between individuals from population 1 and individuals from population 2. As a result, any traits that reinforce barriers to hybridization between these two species should be selected for. So if in their time of separation, let's imagine that, to go back to an old example, population 1 has been selected more towards hummingbird pollination, and population 2 has been selected more toward bee pollination. When they come back together, to avoid these low fitness hybrids, there might be reinforcement for flowers that are really strongly hummingbird pollinated and completely deter bees. So maybe longer floral tubes, for example, in population 1, and in population 2, for flowers growing around population 1, there's going to be selection that really reduces any chance of hummingbird visits and makes these flowers even better for bee visits. So as a result of genetic barriers to hybridization, we can get reinforcement of other differences between the populations that further prevent hybridization and further lead towards these two populations becoming different species. We are going to move on now and talk about the role of polyploidy in speciation. So polyploidy is another genetic change that can create a barrier to hybridization. And it creates a dramatic reduction in fitness of hybrids compared to the original species. So we talked about this a bit at the start, but if you imagine an initial population that is all diploid individuals, so they're all 2N, and they all can mate with each other. If we have a polyploid event, this is an event where usually chromosome numbers are doubled, and so now we have an individual that is 4N. An individual who's 4N, if it tries to mate with an individual who is 2N, is going to create an offspring that has low fitness, because that offspring is going to have chromosomes that don't pair correctly during meiosis, and so it will be bad at making pollen and bad at making ovules. So I'm going to say this makes a low fitness offspring. So what this means is that when plants produce polyploid individuals, they almost immediately become largely genetically isolated from the progenitor species. So in this case, the 4N plant becomes almost immediately genetically isolated from the original diploid plants. Not only do they become genetically isolated, but the process of doubling chromosome numbers changes gene dosage. It changes the amount of mRNA from each gene that's made, and that can change traits. It could affect things like flowering time, or the size or shape or color of flowers. And so, simultaneous to becoming genetically separated because of problems with meiosis, we also have frequently phenotypic changes, changes in appearances, which can lead to the plants having different pollinators or being pollinated at different times. So we can get multiple barriers to hybridization forming at once when polyploids form, both genetic, post-zygotic, and also pre-zygotic barriers arising. Let's look at an example. This is Stephanomeria malheurensis, or wirelettuce. This is a plant I think I encountered out in Iowa prairies, at least a similar one. And it's a plant that has undergone polyploidy. And so here's the original species, Stephanomeria exigua. And after polyploidy, we get this new species, malheurensis. And you can see that in addition to a change in the number of chromosomes, there's also a change in phenotypic traits. Specifically for malheurensis, there's a change in seed size, And more importantly for our purposes, the flowers of S. exigua are primarily outcrossing. So they are large, they are attractive to pollinators. A change that co-occurs with the polyploidy for malheurensis is that these flowers are now primarily selfing. So they are smaller flowers, they are less showy, and they are more likely for pollen from their own stamen to fertilize their own ovules. As a result, self-pollination is itself a barrier to hybridization, because if this plant is not distributing much of its pollen, it is unlikely to pollinate flowers of exigua. So we have multiple barriers then to hybridization between the progenitor species. Let's say, I don't remember for sure, but let's say it's diploid, and the polyploid species, and I would need to check this, but let's say it is 4N. We have both genetic and we have pre-zygotic barriers because the pollination system has shifted. I had planned to address adaptive radiation in this lecture, but because this lecture has gone long, I'm going to hold off on this and see if it fits maybe somewhere else in the remaining lecture. So we'll put that on hold. Let's end with some summary of important points from what we did cover today. First, we talked about the fact that angiosperms are better able to outcross with distant plants, and they are better able to direct where their pollen goes. This means it's easier for them to exchange traits with other plants in similar niches, and it's easier then for species to diverge and take advantage of different niches. Second, we talked about that angiosperms have shorter life cycles. This includes both a shift to being annuals or shorter-lived plants in some cases, but it also refers to the fact that the gametophyte stages happen much more quickly in angiosperms. This creates shorter generation times and more opportunity then for similar amounts of selection to result in greater changes in traits. I'm just going to put an arrow to indicate more changes. And then third, we've talked a lot about different barriers to hybridization that would allow plants that have started to diverge in some traits to have ways such that they don't exchange pollen, or their pollen is not as successful in fertilizing ovules, or that ovules fertilized by foreign pollen are not good at either germinating or the offspring plants have low fitness. As a result, we have pre-zygotic and post-zygotic barriers to hybridization. When these occur, then they are going to reduce either the formation of hybrids or the ability of hybrids to pass down their genetic traits. And that will allow two incipient species to continue having separate genetic trajectories and hence to becoming fully separate species. And finally, we said we'd put discussion of adaptive radiations on hold for a different lecture.